Hi everyone and welcome to week 8. The lesson for this week is about Staphylococcus and Streptococcus, and we're going to learn the different tests to identify them. So Staphylococcus and Streptococcus are two of the most important genuses of bacterial pathogens that cause infections in humans. So you've heard of staph infections and you've heard of strep throat. So those are examples of the infections, but there are other examples as well. So both staph and strep are both gram positive. So that means if you did a gram stain and looked under the microscope, they would appear purple. And they're both coccus shaped, so meaning the round shaped cells that you would see under the microscope. But the difference that you would see if you were to look at them is that staphylococci are arranged in clusters. So the word staphy in Greek means grapes. So you can think of a cluster of grapes is what the staphylococcus looks like under the microscope. And streptococci arranged in linear chains. So you can see in the picture here of strep, the round circles are arranged in a chain or a strip. So to remember that, you can think of strep strip. They form a strip of the circles. So both staph and strep infections are very common in patients. So a test to be able to distinguish staph from strep is the catalase test. And the catalase test involves mixing a little bit of bacteria into hydrogen peroxide. And Staphylococcus is catalase positive, and that means that it produces the enzyme that breaks down hydrogen peroxide into water and oxygen. And as a result of that reaction, you would see bubbles. And you see that in everyday life when you use hydrogen peroxide on a cut on your skin. You see that bubbling because there's staph epidermidis on your skin. Then on the other hand, Streptococcus is catalase negative. It doesn't produce the enzyme, so there's no change and no bubbles. So I'm going to give you a way to remember this, and I'm going to give you a lot of tips for remembering the outcome of the different tests. So for this one, you can think of a friendly staff of nurses in a hospital. So think of staff is bubbly. Now we're going to talk about Staphylococcus and some of the major species of staph and some of the tests to distinguish between those species, and then after that we'll do the same for strep. Staphylococci are normal inhabitants of our skin, our mouths, nose, and throat, and they generally just live there without causing any harm, but they can cause disease when they penetrate the skin barrier or the mucous membranes. And some species can cause very severe infections, which we will talk about. So there are 3 major species of Staphylococcus that we're going to talk about. The first is Staph aureus, and this is a really dangerous one, highly pathogenic. And one of the reasons it's so pathogenic and dangerous is because it produces toxins. And some serious infections are associated with Staph aureus. So serious, severe skin infections can be a result of it. It can cause gastrointestinal infections, and toxic shock syndrome also can occur from Staph aureus if it gets in the bloodstream, and the patient can be septic. The second group is Staph saprophyticus, and it's actually a normal, natural, normal flora in your body. But if a person is immunocompromised, or if the Staph saprophyticus is in the wrong location, it can be opportunistic and cause an infection. So an example of infections commonly caused by saprophyticus include urinary tract infections. And then Staph epidermidis also is usually not pathogenic. It's usually normal flora, but it can be an accidental pathogen. So for example, infections that are associated with from medical devices like catheters. If staph epidermidis gets in the wrong location, it can cause an infection. Now we're going to get some really gross pictures in this lesson, and these here are to show you what makes Staph aureus so much worse than the rest. So I told you Staph aureus is a highly pathogenic and dangerous bacteria, and this is showing you and telling you what it is doing to your patient. So it frequently causes abscesses and local pus lesions, and there are metabolic end products that it produces that are involved in its pathogenicity, meaning what makes it so dangerous. So one of those is coagulase, and what that does is it causes the blood to clot. Another is leukocidins, and they destroy white blood cells. Hemolysins destroy red blood cells, and then DNase, lipase, etc. Break down the DNA in cells and break down lipids in cells. So these are the results of Staph aureus and what it's doing to the patient. Since Staph aureus is so dangerous and so pathogenic, we need ways to distinguish it from other species of staph. So there are several tests that help us to do this, and I'm going to talk about all of them. But the first is called the mannitol salt agar test, or MSA plates. Now, MSA plates are these red plates that have salt in them, and they're selective in that only salt-tolerant organisms are able to grow on them. So all three strains of staph can grow on them. But they're also differential in that there is a color change if the species is able to ferment mannitol. So, as you can see on the picture here, the species that can ferment mannitol change the color of the plate from red to yellow. And the two that are able to do that are Staph aureus and Staph saprophyticus. So there's a trick to remember which species ferment mannitol on an MSA plate, and you can use the letters MSA to remember. So think of M, a mannitol plate, S, saprophyticus, and A, aureus, can ferment on a mannitol plate. Now another test to distinguish the dangerous Staph aureus from other staph is called the coagulase test. And remember, I told you one of the things that Staph aureus does is it causes blood to clot or coagulate, and that's because it's the only staph species that produces the enzyme coagulase, which is the enzyme that causes blood to clot. So if you do this coagulase test, it's using rabbit plasma, and you take some of your unknown bacteria and put it in a rabbit plasma, and if it solidifies or coagulates, you know that you have Staph aureus, and you can see that in the picture. The sample in the picture is solidified or coagulated for Staph aureus on the top there, and then Staph epidermidis and Staph saprophyticus would not coagulate. It would remain liquid in the tube. Another test to distinguish the dangerous Staph aureus from other staph species is the hemolysis test. And this is a test using blood agar plates, which contain 5% sheep blood. Now remember, I told you that Staph aureus breaks down red blood cells, destroys red blood cells, which is one of the things that makes it so pathogenic. So when you do this test, you can determine if the species is beta hemolytic, which means a beta hemolytic organism, by the beta symbol on the plate, it's a clear, transparent area around the culture because all the red blood cells there have been destroyed. So if you see that on a plate, you would know that you have Staph aureus, because that's the most pathogenic one, causes the most breakdown of blood cells, complete destruction of red blood cells. Then there's also alpha hemolytic, which is a partial breakdown of blood cells. So for that one, you would see a change in color to kind of a greenish brown, which is partially breaking down the blood cells. And then there's also gamma hemolytic, which is no breakdown of the blood cells. So you really see no change on the plate. So the species that are gamma hemolytic are Staph epidermidis and Staph saprophyticus. So the way to remember that Staph aureus is beta hemolytic, think of beta is bad. That's the worst one. It causes complete destruction of the red blood cells, which is what makes it so pathogenic. So think beta, bad. So if you determine that your patient's infection is not Staph aureus, then you're going to need more tests. Now let's say you determined that your patient's infection is not Staph aureus, but you still need to distinguish what it is. So here's another test to distinguish Staph saprophyticus from the others. It's called the Novobiocin test, and it uses an antibiotic disc, which you can see in the picture here. So Staph aureus and Staph epidermidis are sensitive to that antibiotic, meaning that if you put that antibiotic on the plate with those organisms, you're going to see a big zone of inhibition, or a big circle, as you can see in picture B here, a big circle or area where the Novobiocin kills the bacteria around it. So that area where there's no bacteria growing, that's the zone of inhibition. But Saprophyticus, on the other hand, is resistant to Novobiocin. So as you can see in picture A, there's no zone of inhibition, and the bacteria Saprophyticus can grow right up to that antibiotic disc. So a trick to remember this one, think of tree sap. Sap is sticky. So Saprophyticus, you can see it growing and all the way up to the disc and almost sticking to the disc. So think sap is sticky. Now let's move on to Streptococcus. We'll talk about some of the major species of Streptococcus and the tests to be able to distinguish them. And just a reminder again about the shape of Streptococcus. If you look at it under the microscope, what you would see, remember gram-positive, cocci shaped, but the difference in Streptococcus is it forms the linear chains. So remember to think of strep, strip, a strip of bacteria. Now, Streptococcus is also responsible for a lot of serious infections. The genus Streptococcus is responsible for more infectious diseases than most other genuses. And Streptococcus are classified by two major methods. The first is hemolytic activity, which we also talked about for Staphylococcus. Remember that it means the breakdown of red blood cells. And remember that there were three types: beta hemolytic, and remember beta is bad because beta causes complete destruction of the red blood cells. So that makes it the most pathogenic. Alpha hemolytic is the partial breakdown of red blood cells, still bad, but not as bad as beta. And then gamma hemolytic is no breakdown of blood cells, no change. And then the second method of classifying Streptococcus is by their serological classification, which is also known as the Lancefield classification. And what does that mean? It means that they're classified by the antigens on the surface of the bacterial cell. And remember that antigens are what is on the surface of the cell. It's what's recognized by your body's antibodies when your body is attacking to try to protect you from an infection. So those antigens are on the surface of the bacterial cell, and that's one of the ways of classifying the bacteria. And there's 20 different groups of Streptococcus, and they're designated each with a letter. Groups A, B, C, and D. D is now known as Enterococcus. They all have species that cause human infections. And today we're going to focus on A, B, and D, or Enterococcus. So if you thought the staph pictures were bad, now take a look at what strep can do. Particularly Strep pyogenes, which is a highly pathogenic bacteria in group A of Streptococcus. But its severity depends on where it's located, whether it's in the bloodstream, being septic, and whether the bacteria are producing toxins or not. So it can be mild. It can be as mild as a strep throat, or it can be very severe. There's streptococcal toxic shock syndrome in the bloodstream. There's cellulitis, which is a severe, fast-spreading skin infection. Necrotizing fasciitis, which is a flesh-eating disease that destroys muscles, fat, and skin tissue if it enters through a wound. And that's what you can see in this picture here. So strep pyogenes, highly pathogenic, especially dangerous in some of these severe diseases like this one, necrotizing fasciitis. And then in group B, that's Strep agalactiae. That can normally be found in the vaginal mucosa, but it can cause neonatal infections. And then Enterococcus, which was formerly known as group D, that's E. faecalis. That's normally harmless. It's found in our gastrointestinal tract and in feces, but it can cause infections in the heart, can cause urinary tract infections, and infections in the bloodstream, such as if it gets introduced into the bloodstream, if there's an intestinal laceration, for example, during a surgery. So remember I told you that hemolytic activity is one of the ways we characterize Streptococcus species. So the hemolysis test can be used to help you determine what type of Streptococcus species you might see in an infection. So Strep pyogenes and Strep agalactiae are beta hemolytic, so complete destruction of the red blood cells by those two species. But then E. faecalis is alpha hemolytic to gamma hemolytic, depending on the strain. It can have little to no hemolytic activity. So just like we did for Staphylococcus, we can use an antibiotic disc to distinguish the species of Streptococcus. But we use a different kind of antibiotic disc. So remember which antibiotic was used to distinguish staph strains. It was novobiacin. For strep, we're going to use bacitracin. And you've heard of bacitracin before, like in triple antibiotic ointment that you can buy over the counter. So bacitracin is a type of antibiotic, and you can see in the picture here, it's a disc that can be put on the plate to see if the strep bacteria is sensitive to it or resistant to it. So strep pyogenes is sensitive to bacitracin. So you'll see that zone of inhibition because bacitracin kills the bacteria around it. Strep agalactiae and E. faecalis are resistant to bacitracin. So you'll see no zone of inhibition. It's able to grow right up to the antibiotic disc. So a trick to remember which strep is sensitive to bacitracin, it's strep pyogenes. So think of bacitracin eats pie. So bacitracin destroys or kills strep pyogenes. Bacitracin eats pie. Another test to help distinguish the Streptococcus species in your patient's infection is the Bile Esculin test. And only one species of strep, E. faecalis, is capable of hydrolyzing esculin. So when that happens, the products react with ferric citrate in the bile media, which causes insoluble iron salts that turn the media blackish brownish. So as you can see in the picture here, you can see the uninoculated tube, which shows you what it looks like starting out, the color that it looks like starting out, and then the positive tube there is inoculated with E. faecalis, which turns it to the dark brown color. So it's the only, E. faecalis is the only species of Streptococcus that turns it to the dark brown color. So a trick to remember which strain is E. faecalis is found in the gastrointestinal system. So faecalis, think fecal, think dark brown. So between those tests that I've described, you would be able to distinguish between the three major species that we're talking about: Strep pyogenes, Strep agalactiae, and E. faecalis. But in the case that additional testing is needed, there's other ways to distinguish them as well. One other way is by their halotolerance or their salt tolerance. So Strep pyogenes and Strep agalactiae are not halotolerant, but E. faecalis is halotolerant. So what does that mean and how can we test it? It means that E. faecalis can withstand a higher salt concentration. So if all three of those are streaked onto mannitol salt agar plates, only E. faecalis is able to grow.